Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Fitria binti Ahmad Munir and my matrix number is 23DBT 20F1036. Today I want to show you how I perform a lab simulation about strict plate tasks. So for this lab simulation, the purpose is in order to perform many of the tests necessary to identify microorganisms. It is essential to isolate the organism in pure culture. Remember, biochemical tests are only valid when a pure culture is used. A common method to isolate a microbe from a mixed culture is by tracking the culture on solid medium such as agar. Growth of the culture on a solid medium results in colonies, each of which originates from a bacterial single cell. Thus, the colony is a pure culture. The colony can then be restructured onto a new plate or used to inoculate liquid medium, both of which are now pure cultures. So next, there's the steps for this lab simulation. First, sterilize the loop in the flame and allow it to cool. Second, pick an isolated colony from the agar plate and spread it over approximately 1 per 4 of the plate using close parallel streaks. Third, flame and cool the loop, which is easiest way is to gently touch it to an uninoculated edge of the agar. Fourth, turn the plate 90 degrees and lightly sweep the loop 1 to 2 times through the inoculated area, then straight into the next quadrant without overlapping previous streaks. Fifth, flame and cool the loop. Six, repeat the step 4, streaking into the third quadrant of the plate. 7. Flame and cool the loop. 8. Repeat the step 6 and streaking the remainder of the plate. And lastly, incubate the plate under appropriate conditions. So, can start the lab simulation now. So first, sterilize the loop in the flame and allow it to cool. So then pick an isolated colony from the agar plate and spread it over. Okay. Then flame the loop again and allow it to cool. Then turn the plate 90 degrees and lightly sweep the loop 1 to 2 times through the inoculated area. Then streak into the next quadrant without overlapping previous streaks. Right? Okay. Streak straight into the next quadrant. Okay, like this. Okay. Then sterilize the loop again and allow it to cool. Then repeat the step for again. Okay. Spread to the next quadrant. Okay. And flame it again to prevent a contamination. Then allow it to cool. Then repeat the step 6. Okay. Okay. Now place the plate in a incubator set at a temperature of 37 degrees Celsius and wait for 24 hours to see the result. Okay, so this is the results for this strict plate test lab simulation. So that's all for this lab simulation. Thank you.